Here's a little video on point mutations. To start off with, we must find our DNA coding strand running 5' to 3' and align it with our DNA template strand running 3' to 5'. This allows RNA polymerase to read our DNA 3' to 5' and make it 5' to 3'. We then move into translation, where we produce our amino acids and eventually a functional protein. But first, we must find the reading. Now that we've found our reading frame, we can use a codon chart to find our start codon. And we see that our RNA strand codes for 4 amino acids, beginning with our start codon, methionine, then valine, then serine, lysine, and finally our stop codon. Now we're going to use our coding strand for the next few examples where I'm going to induce some point mutation. First, I'm going to do a missense point mutation where we have a transition changing our G to an A. A missense mutation changes the amino acid and in this case causes serine to be changed to asparagine. This may alter our protein function or cause this protein to be non-functional. If the protein function is altered in a species that causes an adaptation fitness outcome, it will be selected for in future generations. We could also observe a neutral missense mutation where it does not affect the protein function. Next, I'm going to do a nonsense point mutation where we have a transversion changing our A to a T, which gives us uracil in RNA. A nonsense mutation is one where an amino acid is changed to a stop codon, and in our case, lysine. This will likely stop transcription, terminating the gene from turning into RNA, causing the protein to be non-functional. Thirdly, we are going to induce a silent mutation. A silent mutation is one where the genetic material is altered, but still codes for the same amino acid. In our case, we see a transversion where we change our C to a G. So even though our codon GUC was changed to GUG, we still end up with valine as our amino acid, resulting in the same genetic sequence. This will likely go unnoticed and will not alter our protein function. Lastly, we are going to go through a read-through mutation. This is where a stop codon is changed to a coding codon for an amino acid, resulting in a longer protein. In my example, we see a transition where T is changed to C. This causes our once UAA stop codon to change to glutamine. We then see that the amino acid continues, adding serine and threonine, when we do not run into another stop codon that we can see. Since this results in a longer than normal protein, this will typically cause the protein to be non-functional.